So as Maria Alejandra said, I will try to give you it'll be like a primer on bacteria and archaea, so that you're all studying, I guess, microbiology or related fields, so you really can look up most of this information, but just some of like a different take on what I think is really wonderful about these microbes, as I write there, wonderful microbes, bacteria, and archaea. And I guess the reason we are all here is because you guys are all involved in doing this microbe directory, right? And I think this is a magnanimous challenge, and you will see why. And you have probably found this out as you're working on it. Trying to really assimilate and gather all the information on microorganisms is not trivial. And it is very complex, and you will see why if you haven't already found out why. I just have here like a snapshot of the microbe directory, and of course here some of the groups that you're working with, from viruses to eukarya. And so what I will focus on now is essentially bacteria and archaea. And I have this picture there, which you already all know what that is. This is our little island in the universe. This is planet Earth, of course. And what is fantastic about this planet is that it is a host to so much wonderful life. And most of that life on the planet is prokaryotic or bacteria and archaeal in origin. And of course, the planet was not always that way, as you will know. This is just a graph taken from the internet, I think from a NASA site. But obviously, planets started to, when the planet started, let's see if I can put a laser pointer there. That when the planet started, of course, Earth was really an inhospitable place to live in. There were no conditions for life at all. And then slowly, over billions and billions of years, this changed. And microorganisms started to evolve. And there's still a debate on how these first actual living forms originated, and that has not been solved, although more and more there's indications of what was going on. But of course, from unicellular, let's say, simple forms of life, we then developed into more complex and eukaryotic species as we see today. And obviously, this is accompanied by changes in the planet. And so the planet has also evolved throughout the years. And this is just a timeline that you can also find on the internet, which I think is very illustrative of how things have changed. And so here you can see that once the world or planet Earth was formed, then it took about several billion years for the first forms of life to come up, and we believe those were prokaryotes, and those are obviously, as you know, single-celled small organisms that lack a nucleus. And then things started to change from then, and there's this big event here called photosynthesis, in which microorganisms were capable of taking in light to produce energy, and CO2 to produce carbon compounds, which we need to grow on, and from there release oxygen. And in doing this process, they absolutely changed the planet, and it became a planet that has an oxygenic atmosphere, which is the planet we know now. And so it is very important for you to know that the planet we know today has nothing to do with the planet when the first forms of life came to be. In other words, we have a planet that is a living planet that has been changed both because of geological and other things that have impacted our planet, as well as by the microbial forms that inhabit it. And of course, we have evidence of this as evidence of the atmospheric oxygen that we have, which was called the Great Oxidation Event. And during that Great Oxidation Event, most of the living organisms at that time died off, because as you probably know, and I'm not sure, oxygen is very toxic to most organisms. We have mechanisms to avoid oxygen toxicity in our cells. And evidence of microbial life is in many places, if you care to look. And this is a wonderful location. These are called the White Cliffs of Dover, which are in the UK. And so you can see, as you're coming up to the shore, you see these wonderful cliffs full of white chalk material. And that chalk material is the remnant of deposits in the ocean floor from millions of years before, of these tiny little algal organisms called Emiliana huxley, which is a coccolithophore, and it just forms these clusters as you see here, and then through the act of millions of years, it deposited in the seafloor, and then obviously through other geological events, the seafloor rose up, and this is what we see today. And so there is evidence throughout the planet, and this is what people look for when they're trying to find fossil evidence of how long ago these microorganisms were alive. But this also indicates that microbial forms have been shaping this planet for millions and millions of years. And of course, this interest in trying to see what was there evolved slowly over time. And now we're jumping to the 17th century, and many of you might have heard of this person who's called Anthony van Leeuwenhoek. I don't know if I pronounce it right, but this was a Dutch merchant, and he worked with, he used to sell fabrics. And in his interest in trying to identify the best fabrics, he developed this tiny little implement, which actually works as a tiny microscope, and you can deposit a tiny droplet of a sample. And through this tiny little microscope, he was able to see the first observations of these tiny little microscopic forms, which he called animalcules, and this he published. And of course, this is why we now credit him with one of the first observations of these microscopic figures, beings. And it was interesting because he could pick up, for example, water from the channel, and he could see that these things were there. He could take samples from his own saliva, and he could see evidence that these things were moving around. And then he also knew that if he took some hot beverage after he had taken the sample, then many of these living forms would sort of like stop wiggling around. And therefore, he imagined that these must be living forms, of course. And then we can jump a couple of centuries again. And this is now in the 19th century, and we come to some of what we call the fathers of microbiology. And this, we have to mention, of course, Louis Pasteur, who was a genius in his own time and a person who worked in several different fields, not only microbiology, and is well known, of course, for fermentation and for wine development, as well as for pasteurization. But in the field of microbiology, he was also a crucial figure in terms of doing a small little experiment, which is depicted here to the right, in which he basically what he did is he took some what we would call now medium, and he autoclaved it, and he either left it open to the exposed to the air, or he closed it off with a funnel like this, such that oxygen could still come in, but there would be like no pollution, no air and particles that would come in. And the one that he closed off to the environment remained sterile, whereas the other one was contaminated. And with that, he dispelled the original theory that people had at that time that there of spontaneous generations, that life just spontaneously evolved out of nothing at all. And so he also dispelled that theory and confirmed what we now have called the germ theory, which basically illustrates that there are microorganisms there that can cause different diseases and can be used for different purposes. So Louis Pasteur was fundamental in changing our perception of what microorganisms can do. And this is Robert Koch, who is another one of those fathers of microbiology. And he worked simultaneously around the same time as Pasteur, but in Germany. And he was very meticulous in his methods, and he, in fact, was capable of isolating quite a few microbes. And in this case, he's very famous for isolating the microorganism that causes anthrax, which is Bacillus anthrax. And it is said that, in fact, both of them did not like each other very much, as usually happens when people are competing against each other. But Robert Koch is also very famous for, actually, as I have here in the title, teaching us how to make pure cultures, which is the way many of us in microbiology work today, and is the reason why we are having now more and more problems in trying to understand that in nature, for example, microbes do not really occur in pure cultures or in isolation, but probably in communities. And that's why we're now trying to shift our understanding from understanding pure cultures to understanding communities of microbes, which is quite a challenge. And so why were these scientists so interested in these microbes? Because microbial diseases, and here I'm depicting just two of these cases, have accompanied humanity throughout their history. And to the left, you can see a picture from Bruegel, which is a painter who basically depicted what we believe was the disaster of the bubonic plague, which was caused by Yersinia pestis in Europe, in which approximately 1/3 of the population in certain localities died, and it's a beautiful picture. You can go and check it out sometime to see all the tiny details of these death figures coming around and how people were dying, because probably this is what they felt when they were succumbed by this disease. And then to the right, I have a picture of what was actually probably closer to what is happening today, which was the 1918 flu pandemic. And you can see here this woman saying, or this person saying, wear a mask or go to jail. And I just put it up there because this is reminiscent of what is happening today. And so these things come and go throughout humanity. And again, we have to sort of relearn how to do certain things. But the essence of this slide is to show you that, in fact, humanity has been shaped by these pandemics and by microbial diseases, which is the reason why, of course, we have such a robust immune system. If we didn't have an immune system, we would have gone extinct long ago. And so we have co-evolved with these microbial partners. And along the way, we should not forget that many people were also interested in the environment, and they started to study microorganisms in the environment. And one of these is Bayerink, and he was very well known for identifying microbes involved in nitrogen fixation. And that is relevant, of course, to how we carry out agriculture. Without microbes that fix nitrogen, in other words, that can convert nitrogen into forms of nitrogen that we can then assimilate. For example, we cannot fix nitrogen. We all depend on these nitrogen fixators in the environment. Of course, now there's chemical methods to produce the fixed nitrogen, and so we can circumvent the use of some of these microbes. But at that moment, of course, it was a breakthrough discovering. And on the left, you see these tiny little nodules that form on certain plants. And in these nodules, you can find these nitrogen fixing bacteria that actually provide nitrogen to the plant in a sort of symbiotic relationship. And then another environmental microbiologist that is very well known is called Vinogradsky, and he constructed what you see on the right, which is called a Vinogradsky column. And you can do this. You can find the recipes on the internet, but basically you can just take sludge or mud or particles from certain environments, and you can layer them in a tube that has no oxygen on the bottom, and the microbes will sort of form layers depending on how they can utilize nutrients in the different layers. And so you'll have an oxygen on the top, an oxygenic on the bottom, and then these organisms will sort of diffuse to find their own places and accommodate to use the nutrients that they find within a certain location. And of course, what this is illustrating is the fact that in the environment, you have all these microorganisms utilizing nutrients in a sort of and using these nutrients among each other. And so he was very important in identifying microorganisms for geological cycling of nutrients in the environment, which is the way the planet is sustained. And so in fact, our planet is sustained mainly because we have all these microorganisms doing all this work for us. And they are the primary producers to a large extent. And then we benefit from all this material that is being utilized and being produced and converted by these microbes. But of course, along the way, people started to identify bacteria in different places and recognize that they were relevant and important. And there became the need to sort of classify this. And so humans, we have a need to classify things. And we like to say, things are good or bad or right or white and black or up or down. And so we need to understand things by classifying them. And this is why, of course, we started to devise mechanisms to establish relationships among the organisms that we are identifying, which is what we call a phylogeny. And on the right, you can see a very crude drawing done by Charles Darwin in his book, The Origin of Species. And here you have the origin of what a tree might look like, which obviously has changed throughout history. But then later on, this investigator from Germany called Haeckel, who was very interested in beautiful drawings of microorganisms. Some of his theories have not held through, but still he was a firm believer in evolution as proposed by Darwin. And he, of course, constructed this beautiful geological tree of what he calls humanity, which he has some of the microbes in the bottom part of the tree. And then this, of course, has evolved over time. And then at the beginning of the last century, we have a tree looking like this, in which the major, more important forms are attributed to plants fungi, and animals. Whereas in the bottom, you see what we call protists, which are single-celled, like protozoa, eukaryotes. And in the very bottom of the tree, you find what they call monera, which are the microorganisms or the prokaryotes. And this has changed substantially in the past years because you well know we now have other tools with which we can do phylogenies and establish relationships among organisms. And here's just a diagram illustrating how sequencing has changed over time. And at the beginning, we had gel-based sequencing, and we'd had to do everything manually. Then this was a first-generation sequencing. We did capillary sequencing, and now we have next-gen and third-generation sequencing. And just the amount of information in the databases has just exploded. And with that comes the challenge of what to do and how to interpret all that information. But needless to say, this has changed the way we view this whole organization of life on the planet. And this is what we now call the tree of life. And one of the big breakthroughs came around in the 1970s, late 1970s, with the work of this researcher, Carl Woese. And he was the first one to actually look at molecules of RNA. In that case, in his time, he actually looked at the RNA instead of sequencing the DNA, because sequencing of DNA was not possible at that time. And with that information, he defined that there should be 3 large kingdoms or domains of life, which he called, well, at that time, Eukaryotes and Archaea, which were distinct from eukaryotes. And this little square here on the side just illustrates that most of the macro life that we know, plants, animals, and fungi that we can see, really occupy a very small part of that tree of life. And of course, there were many other researchers who contributed to our idea that really the vast majority of the diversity in this planet is occupied by these 2 groups, which are the bacteria and the archaea. And to the right, I have a new let's say, tree of life, which was published in 2016. And many of these, so all of these branches represent different groups of bacteria and here of microbes, of organisms. And here, of course, you can see some of the large groups like the Proteobacteria, which you probably have worked with. There's others here like the Bacteroidetes and Microbe, Actinobacteria, Chlorophyll, Firmicutes, etc. But what this tree really did was that they used lots of sequence data. And so all of these red dots that you see here do not correspond to isolated microbes, but rather to information that you gather by sequencing. And so all of these red dots indicate branches in this tree of life that do not have any cultured representatives. And it is interesting because really, while we can get all this information from sequence data, really most of the information on how microbes live and are organized in groups, or what it is that they're actually doing in the environment or in the human, to a large extent depends on whether or not we can culture them and do experiments on them. So lots of people are actually trying to culture some of these representatives because it would really be a worthwhile endeavor. And then the other thing interesting about this is this group here called CPR. Let me see if I have it in the, I don't know. So this group called CPR, which is called the candidate phyla radiation. And really they discovered a whole new branch within the bacterial cluster, within the bacterial kingdom, that is distinct from the rest of the bacteria. And they're small cells, probably substantially different from the rest, that merit having their own sort of division within the tree. And so in essence, what we know now, based on the work of myriads of scientists and explorations throughout different environments, is that we really live in a microbial world. We're immersed in microbiology, and microbes surround us. And as you well know now, because of this pandemic, it can have very harsh consequences. But again, most of the microbes are really non-pathogenic and really do not cause us any harm. And now I just talk a bit more about specifically bacteria and the archaea. And this is just an expansion of the tree I showed you previously from 2016. And just so that you can see, these are the groups that belong to the bacteria. This is the candidate phyla radiation, which I mentioned. And then up here is the archaea, which I will mention briefly later on. But most of the diversity and most of the diverse forms of life, we believe, are encompassed by the phylum, by the kingdom bacteria. And so why are bacteria, what are bacteria? And you probably all know because you're all microbiologists, I gather. But anyway, this is just a very simple figure. And of course, there's always exceptions to the rules, but bacteria, both bacteria and archaea, are distinct from eukaryotes in that they are single-celled microorganisms that lack a nuclei. And that was the definition, and that was why they were defined as prokaryotes originally. And now there's people who are really against the notion of having a prokaryote nomenclature, and you will see why, and that is because archaea and bacteria are quite distinct groups in and of themselves. And so some people argue that we should not use the word prokaryote anymore. But still, it is very useful in the sense that it defines cells that are free living, usually exist as single cells, have no nucleus, have therefore all their components swimming around in the cytoplasm. And that, of course, generates a substantial difference with eukaryotes and the way in which they produce many of their components inside the cell. So their metabolism is distinct from those of eukaryotes. And of course, here I have some other distinguishing features. Some of them have these things like called flagella that allow them to move around. They, of course, have proteins on their outer surface that allows them to interact with other species and to sense their environment. Then, of course, they have a cytoplasm. And, of course, they have membranes. And the difference, as you will know, between gram-positives and gram-negatives is that one of them have one membrane and the others have two. And usually they have cell walls, which is like this firm cellular matrix that gives them the shape and the firmness. But as always, there's always exceptions. And here I have a small little picture, what is called a mycoplasma, which are really tiny cells that usually are parasitic and cause diseases. And these cells are bacteria, but they have no cell wall. So what I would like you to take away is that even though there are some generalities, there usually are exceptions to the rule. And so once we know we have bacteria, of course, people like to classify them, as I have already mentioned, and it's important to identify them. And there's a whole big field of trying to do phylogeny and identification and classification. And you wonder why, and you are all working with the microbe directory, specifically with particular species. And that is the result of all this classification and identification. And the reason we like to do this is because it gives us a sort of a broad idea of what sort of organism we're working with. And so it is relevant in the sense that we can organize our work in our minds and know how to deal with particular living entities. And so ways of classifying involve, for example, morphology. And even though all microbes are very similar, they do have distinct morphologies, as you can see here. Then this was a huge step when Dr. Graham invented the Gram stain, in which you can actually discriminate between what we call gram positives and gram negatives. And here you see a gram positive bacterium that stains purple, while gram negatives are counter stained, and there you see a reddish color, and this is like an E. coli. And so you can distinguish microbes, at least bacteria, into large groups that we call gram-positive and gram-negative. But again, as I mentioned, there are exceptions to the rule. And even though you think that this is a group that is a gram-positive group, sometimes they will not stain as you think they should. And so you have these gram-variable things. And then there's other way of distinguishing microbes. For example, you can look at their metabolism, and there's tons of assays that you can run in terms of what sort of sugars they ferment, what sort of products they produce. And that, of course, gives us a basis for distinguishing between microbes based on metabolism, which is perhaps better than, for example, just doing a gram stain. And nowadays, we also distinguish microorganisms and do lots of establishment of whether or not you have a new species, for example, based on cellular components. And of course, the use of DNA has been widely used nowadays to do this classification, mostly the 16S ribosomal unit or the small cell unit. But in order to establish new species, you have to take into account different molecules within that bacterium. And this slide I just have here to illustrate that bacteria can grow on all sorts of different environments. And so you see, for example, this Francisella, which causes diseases, growing on a macrophage. But you can also find bacteria in really inhospitable places like hydrothermal vents, and these are called extreme environments, of course. And you can find them everywhere. You can go to a forest and you can look at your hands and your mouth, and you're full of bacteria. So it's just to illustrate the versatility that they have in terms of growth and nutrition. And it is confusing sometimes, but bacteria, of course, vary depending on how they grow and what sort of nutrients they utilize. And this is just to illustrate some of the categories that you can find when you're looking at bacteria. And of course, the most widely known are perhaps the phototrophs, such as cyanobacteria. And these phototrophs are really primary producers and important for our planet in terms of because they can actually use sunlight as energy and then use carbon, CO2, and fixed carbon to generate carbon compounds. And so this is important because this is the basis of all life in this planet. You need a source of energy and you need a source of carbon in order to produce all the components that is going to make an organism. And so phototrophs are essentially, they can be autotrophic. In other words, they can fix energy. They can use energy from sunlight and fix carbon, and they can exist without any other help from anyone else. Whereas many of the other organisms on this planet, such as our cells, we are of course restricted to the amount of restricted in terms of the where we get our energy and where we get our food. And so, for example, we are heterotrophs as humans. We cannot fix energy, we cannot fix sunlight, and we cannot fix carbon. So we depend on other forms of life to have done that previously, and then we can utilize that to thrive. But of course, microorganisms can also do that, and there's all sorts of different lifestyles. These, for example, the lithotrophs can actually use inorganic compounds to generate energy, and some of these you find in really extreme environments, like in the hydrothermal vents, for example. And so one of the biggest concerns throughout the history of humanity has, of course, been pathogenesis. And this is why you see in many of the databases an overrepresentation of what we call pathogenic bacteria, when in reality, the majority of bacteria in the environment are perhaps non-pathogenic. And here I just have some pictures of some of these microorganisms that we consider pathogenic. And the reason we know so much about them is because they're relevant for our health and for industry. I mean, not only human health, but animal health, plant health, etc. And so we know a lot more nowadays of these pathogenic bacteria than of environmental bacteria, which are so relevant to the ecosystem. But again, it is important to take into account that pathogenesis is not a yes/no answer, a yes/no solution to anything. Really, there's lots of shades of gray, and that has to do with the fact that we have evolved within this pool of microbes, and you can find microorganisms within an ecosystem that can go from being mutualistic. In other words, they benefit, they can actually benefit both of them from the relationship, to a parasitism, which is what we call a pathogenesis. And so sometimes it is difficult, in fact, to distinguish between an opportunistic pathogen and what we would call a more real pathogen, because many of those perhaps are opportunistic, like, for example, Klebsiella pneumonia. And so some of these that we consider pathogenic, like Klebsiella and E. coli, they are part of our human microbiome. And so you can find them in different locations in your body. And it is just when some of our immune systems are infringed, or something happens or you get a tough cold, or something else happens to your body, that perhaps you get an infection. And so understanding how these pathogens actually cause an infection is also very important to our health and disease. And I have here one example, for example, Mycobacterium tuberculosis. We have worked extensively also with this microorganism, and which I find fascinating, and is difficult to cultivate, and is really one of the organisms in which we have found that can only thrive within the human. In other words, it seems to be like a real pathogen, but it is becoming, it seems to slowly be evolving to be more of a symbiotic microbe, in the sense that if it manages to stay within the human being for a long time, and perhaps not cause so much of a disease, and perhaps in that way thrive and be able to be transmitted to other people. And so microorganisms, we have also many examples of how they can be, in some cases, pathogenic, but in other cases they can sort of attenuate their pathogenesis to see if they can thrive and persist in time. And this Mycobacteria belongs to a large phylum that is called Actinobacteria, which I will mention now briefly, and in particular with respect to the discovery of drugs to control many of these diseases. And of course, you have all heard perhaps of Alexander Fleming, who was credited as the discoverer of penicillin, because he saw some plates on his bench in which he had a fungus growing that actually stopped growth of bacteria. And so in this case, penicillin is produced by a fungus, but many of the natural drugs that we now use to control infections are also produced by this larger group of bacteria that are called Actinobacteria. And this I have here a lovely picture of actinobacterial colonies, which we also call, this phylum is called the high GC content Actinobacteria. And you can see here these lovely columns. And because these colonies are hydrophobic, they produce these tiny little droplets that are full of antimicrobials or secondary metabolites or specialized metabolites as they're being called now. And many of these specialized metabolites are what we now harvest to produce as drugs against infections or other uses in industry. And of course, due to the high use of many of these antimicrobials, we now see a problem of antibiotic resistance. And the question, I'm not going to talk about because that's a huge different area to talk about, but the question is what to do about this, how to actually either discover new drugs, which is very slow and time consuming and costly, or take a lot of care in the way we administer and use antibiotics to sort of minimize the appearance of drug-resistant microbes. And here I just want to mention another important group among the bacteria. It's called the low GC gram-positive bacteria. These are firmicutes, which you probably have heard about. And many of these, some of these cause diseases like Staph aureus, for example, and strep pneumonia. But there's also very important members of this group, like the lactic acid bacteria, which you consume whenever you eat yogurt, and important in fermentation. And then there's other microbes, for example, that form spores. And this group is very interesting because they can form these tiny little spores within the cells, in contrast to spores that are outside the cells. And these, for example, these are spores of this microbe called Bacillus subtilis, which is really non-pathogenic, non-harmful, and has been used extensively in the lab, not only to study spore formation, but to study in the last years biofilm formation, which is also considered to be relevant in many environmental conditions and in diseases. And sometimes we presume that biofilms can be formed in many, for example, medical devices or surfaces, and then they serve as a source of microbes for diseases. And so Bacillus subtilis has been used a lot to study in the lab. But of course, there are also other members of this group that cause diseases like Bacillus anthracis, the cause of anthrax. And then Clostridium difficile, which is one of the microbes nowadays that as people administer antibiotics, especially in the elderly, much of the microbiome is affected in a certain way such that you begin to have a predominance of Clostridium, even though it is part of your microbiome originally. But when you get too many antibiotics, it sort of comes up and becomes a major member that now affects many of these patients. And then members of the Clostridium group are also important in industry, like this acetobutylicum, which is important for solvent production. So you see, many of the group of these microbes, even though they're clustered and they belong to a certain phylogenetic group, they do have distinct functions and differences, even though they're similar at the level of their proteins, DNA, etc. And of course, we shouldn't forget that many of their microbes are important in the environment, and in particular, for example, those that are involved in carbon fixation, which I already mentioned, nitrogen fixation, which is the conversion of nitrogen to ammonia, which is crucial for all of us to live here. And a lot of pseudomonas, even though some are pathogenic, there are some that are important in decomposing material and in nitrogen and carbon cycles. And now I'll briefly talk about the Archaea, because I see it's already a bit late. And so the Archaea is interesting because despite being also a prokaryote, it is distinct from the bacteria. And in particular, there are certain differences, like, for example, their membranes, and the lipids that are within those membranes are different from those of the eukaryotes or of the bacteria, and also some of their components. So in this case, for example, I'm showing here an RNA polymerase in which the Archaea are more similar to the eukarya than they are to the bacteria, which is interesting. And in fact, these are just some of the microbes that you probably might come across, which are part of the Archaea, that are interesting because they tend to have extreme conditions of growth, like the Methanogens and the halophiles. These halophiles can grow in really saline environments, and for example, you have odd structures like this Haloquadratum Wellesley, which actually forms squares instead of being round cells. And then some of these Haloarchaea that can thrive under these conditions, you can see the presence. You can detect that they're there because they form these wonderful pigments in the presence of salt. And one of the marvelous things about, and the things that have changed in the way we perceive how life has evolved, is that Archaea presumably, well, Archaea have been identified to be a much closer relative to the eukaryotes than they are to the bacteria. And so it seems that eukarya probably has evolved from Archaea. And in fact, you can see that many of the proteins have eukaryotic signatures. So these are proteins, and you compare them with eukaryotes, and all these little dots indicate that they're similar. And so in many ways, Archaea are more similar to eukaryotes than they are to bacteria. And that is why some people are actually a bit opposed to the name prokaryotes. And in fact, recently they just isolated, and it took them a long time to isolate a member of this group of Archaea called Archaeota. And here's a picture. And this Archaeon is the one that is the closest relative that has ever been cultured. It's the closest relative to eukarya. So now you can imagine the whole slew of experiments that people can carry out with these culture representatives to see how they actually are similar or different from eukarya, and whether or not this can give us some information into how eukaryotic lifestyles originated. And so I would just like to end illustrating that microorganisms illustrate how how this whole planet evolved in interdependence. We all depend on something else, and as humans, we depend on things that are in the environment as well as in our body. And these relationships can go from mutualism to parasitism. And there's really, it is sometimes very difficult to distinguish between these different states of being of an organism. And in fact, I'd just like to end with this slide showing that due to the characteristics of our wonderful planet Earth, that has given rise to all these fabulous different life forms, is the reason why we have life as we know it right now. But here to the right, I would just like to show you a tree of life as depicted by Kunin and his collaborators, in the sense that what is happening perhaps in the prokaryotes, or let's say bacteria and Archaea, is so difficult to understand because there's lots of what we call horizontal gene transfer. And and what that means is that these organisms can share their genetic material among bacteria or cells that are not necessarily related to them. And this is what is called horizontal gene transfer. There's different mechanisms of this occurring. And that is why sometimes it is so difficult to actually pinpoint what is a bacterial species. And there's a huge area in which people still argue whether or not we should actually call these species and how to distinguish one from the other. But just for the sake of clarity, people still like to stick to the notion that we can distinguish bacteria and call them species, because it gives us a clear sense of what type of work we're doing and who we are working with, and how to deal, for example, with a pathogenic mycobacterium versus an environmental pseudomonas. So it does distinguish certain characteristics that enables us to work and understand what is going on. But it's important to keep in mind that this is really a very mobile world. And in particular, the prokaryotes are changing constantly. And so what we see now might not necessarily reflect what will come in later years, or as you start to work with your own microbes. And so with that, I would like to stop and, thank you, everyone. And if you have any questions, please go ahead.